All right, so here is an example I put together about using ANOVAs. Hopefully this will be a nice way to sort of think about an ANOVA without just looking at the notes and the equations and just kind of thinking about how an experiment might come together. So I did this on the Algae project because this is the research that I do, so I have some real data that I think we can use to illustrate the point. First, to give you a little bit of a background of the project, the picture you see up in the top corner is of all the flasks. These are the ones that we use in our lab, and we do all kinds of experiments to make sure we know the best way to grow the algae, including what kind of media to use, what kind of temperature to use, what kind of pH to use, whether or not they can handle having acid added to them because they're going to see some acid when they encounter the flue gas, and whether or not we can buffer the system by adding, various components. So we do all that stuff in the lab in those flasks. And if you look over to the next picture, it's a smaller version, and we have this trailer unit down in the lab, and it's a smaller version than the big, but it's bigger than the flasks. And then the next picture down is the long rows. Those are out at the Center for Applied Energy Research, so we are currently running those, and so you can see how things keep getting bigger. Now the final picture, which is not a real picture, it's an artistic drawing, but it shows the rows of algae photobioreactors right there at the coal-fired power plant. So again, that's not real, it's just a really good picture, and it illustrates the point of where we are and where we want to go. This is just another picture of the overall process so you can get an idea of all the different pieces that we have to put into it. Right now we're just focusing on what kind of water source makeup to use in terms of this example. So let's look at what kind of water. Okay, so when we're in the lab, we use flasks for our studies like I showed you, and we basically just use tap water for our growth medium. It's easy to get, and it doesn't have to be distilled water, it doesn't have to be anything fancy, so we just use tap water. Now, when we're on site and we have 8,000 liters to fill, tap water might actually get a little expensive. So the questions we're asking are things like, can we use something else, like maybe something from an ag facility or urban river water or even pond water, which actually makes sense to grow algae in these things since in nature that's exactly where algae grows. So the experiment we put together, we did a treatment, which was water source, and we had a total of 4 different water sources. The first being tap, and then something from an ag facility, and then urban river water, and then pond water. And if you wanted to, you could think about the tap being the control because that's typically what we use. And then we were going to try and see what other responses we could get from the other kinds of water. Now we did these with 3 repetitions, and when I say 3 repetitions, that means that 3 of the flasks had the same treatment. So of the 4 treatments and the 3 repetitions, we had 12 flasks total. And the foreshadowing is that this is a one treatment, one way ANOVA. Okay, so before we go any further, we've talked about the experiment, and I want to do a quick flashback. So what's the experimental unit? Yes, it's the algae in the flask. You could go ahead and kind of think of it as the flask too if you want to, except for it's really the algae in the flask. Okay, so what's the response variable? What is the thing that we are measuring? The algae growth. We want to know how much algae is being grown or even the growth rate. We're actually going to use growth rate. Okay, and what's the explanatory variable? Remember, that's the one that we think is going to change the response variable, right? The water used. All right, so here's our results from the experiment. Now, the first thing I did was I plotted the averages for tap, ag, urban, and pond, and I calculated a standard deviation and put the error bars on. So I have yellow arrows pointing to those bars, and that yellow arrows represent the error that we see when we're trying to keep everything the same. Inside of each one of those bars, all of the data, all of the flasks were treated exactly the same. Now, they didn't end up with the same growth rate at the end, but we tried. So with the change in growth rate that we see for the yellow arrows is basically the error that you would expect when you're trying to keep everything the same. Now, in the final bar, which is labeled all, I took an average of everything, and I took a standard deviation of everything. Okay, so now this doesn't just represent the change that we would expect when we're trying to keep everything the same. This one actually represents the change that we would expect to see over all of the treatments. Now, the question is, if the error is bigger when you go across treatments, which is the brown arrow, than what you would see with the yellow arrows, you can assume that there's something happening with the treatments. There is a treatment effect. Now, this is a nice example to use because I think you can see the treatment effect. Tap water is really not performing as well as the other three. So you can see that there's going to be a treatment effect. Something about that water is going to affect those averages, the overall average and the overall error. And the overall error is going to end up bigger than the error that we see inside of the bars that have the yellow arrows. Okay, so now look down at the bottom. This is the output from Excel for a single factor ANOVA. And I've gone ahead and labeled the brown arrow and the yellow arrow. So this ANOVA is going to have two different sources of variation, the between the groups and the within the groups. Now, between the groups is the brown arrow, within the groups is the yellow arrow. And the error that you see is basically quantified by these two numbers next to it. In the column labeled SS, that stands for sum of squares. And it's a quantification of the error that is between the average, overall average, and the individual points. And we're not going to actually calculate it, so let's just leave it at that. Now, remember when we've done other things with error, we wanted some credit for how many data points we had. And in this case, what we're going to use is the degrees of freedom. So in that next column, what you'll see is a 3, an 8, and an 11. Okay, between the groups, basically there's 4 different groups, so the degrees of freedom is 4 minus 1, or 3. Okay? The total at the bottom, there are a total of 12 data points, so the total degrees of freedom is 12 minus 1. Now, the easiest thing to do is just put that number in the middle so that it all adds up. So 3 plus 8 is 11, and those are your degrees of freedom. The next column is labeled MS. That's the mean square. And the idea there is we're going to start with the sum of squares and then divide by the degrees of freedom. So you can see that we get an average type of error. And you get some credit in there for how many data points there are. Okay, so now what we want to do is compare the error between groups to the error within groups. And when you're comparing two things like that, one of the easiest things to do is take a ratio. So that next column you see, which is labeled F, is the ratio of the two. And it's actually between groups divided by within groups. So the top one's the numerator, becomes the numerator, and the bottom one becomes the denominator. So it's nice that they organize it like that. This is also called the F statistic. So we did T statistics, we did Z statistics, and now we're going to do an F statistic. And in this case, unlike when we saw in the regression, Excel goes ahead and gives us the F critical to compare it to. So the F critical that we're going to compare 10 to is 4, which means that our F value is going to be bigger than our F critical, and we're going to go ahead and assume that there is going to be a significant effect. And if we want to use a p-value, you can see that it's pretty small at 0.003, again, a significant effect. Now, remember, I'm just kind of showing you the end result so that hopefully when I show you how to get there, you'll appreciate it. So if that didn't make too much sense, then definitely consult the notes and hopefully it will make more sense. But what I thought would be good is to go ahead and take that tap water out and look at it again. Okay, so here's the results without the tap water. And now my column for all, it's only the average of the ag, the urban, and the pond. Now, take a look at this one. In this case, the error across all three is about the same as the error that you see individually. Now, when you actually do the math and the ANOVA down at the bottom, what you'll see is since you get so much more credit for the within groups because there's more degrees of freedom, it's going to cause the mean square to be such that when you look at the F statistic, it's going to be 3.4. And we're going to compare that to 5.14. So we've got a p-value of 0.1, and remember what a big p-value means? It means basically things are the same. So there's no significant effect of water if we just look at the 3 off-site waters. Okay, so I just want to go through a different example just to kind of give you an idea of another ANOVA that we might be able to use. So instead of just thinking about the tap water difference from when we're dealing with what's going on in the lab to dealing with what's going on site, the other thing is the sterilization. In the lab, we use filters. We just run all the water through a filter and it gets rid of all the bacteria. But when we're talking about 8,000 liters, that's going to be too time consuming. So the question is, what if we use UV sterilization? Well, we wanted to go ahead and test in the lab to see if there was a difference between the two, especially with reference to these off-site waters, because really they could have more bacteria in them than we've ever seen in the tap water. So the experiment we used was actually 2 treatments now. One treatment was for water source, and the other treatment was for the sterilization method. We still had 3 repetitions, and by having 3 repetitions, that means that we still have 3 flasks with the same treatment combination. So we have 4 times 2 times 3, or 24 flasks. In this case, what we have is 2 treatments and a 2-way ANOVA. So just to give you an idea of what the difference is between a 1-way ANOVA and a 2-way ANOVA. So here are our results, and you can see that the ANOVA has gotten a little bit more complicated. For one, it's got samples and columns, so you're going to have to keep track of which one you put in samples and which one you put in columns. It also has something called interaction. This is because there can actually be something that happens when the 2 things get together that make them significant. And then within, it only says within, but that actually means the error. So if we look at our p-values, in this case anyway, we're going to see that everything is not significant. There's no, everything is significantly different because we all have small p-values. But I just want to give you an idea what that looks like. Now, one last thing before we go. What if all the experimental units are not the same? Well, if we don't have experimental units that are all the same, and if we were going to use the algae example, we might say that we were going to compare Scinadesmus algae in one experiment and Chlorella algae in another experiment, then what we would need to do? Well, in that case, what we need to do is block on the experimental units. And a block inside of an ANOVA actually gets treated almost like a treatment. So this slide explains the experiment with 1 treatment, and we're blocking on algae strain, but ultimately we still have 24 flasks. We still end up with 1 treatment and 1 block and a 2-way ANOVA. So I just wanted to draw attention to the fact that within Chapter 12, we're going to be dealing with situations where we have 1 treatment and we use a 1-way ANOVA. We're going to have situations where we have 2 treatments and we're going to use a 2-way ANOVA. And we're going to have situations where we have 1 treatment and 1 block and we're going to use a 2-way ANOVA. Interestingly, the other reality is, since in both of the latter cases we're going to end up using a 2-way ANOVA, sometimes it doesn't quite matter if you treat things like a treatment or a block, as long as you're sure that everything is randomized and all of the confounding is reduced by that randomization. So that's all I have for this discussion. Hopefully that helps give you an example, see some real data, see why we would want to test any of these things so that you can be excited about learning more about ANOVAs during Chapter 12. Thanks a lot.